So is the screen visible? Is the screen visible? All right. So good evening, everybody. Today I'm going to talk about biochemical reaction. Although our flyer said that we'll talk about culture methods also, but that we will take some time later because when I made the PPT for biochemicals, I realized it's going to be a long session. So let's not keep you here for more than 45-50 minutes. So I'm going to talk about the biochemical reactions. Hope you all can see the screen and you can hear me and the slides are moving. I'll just take a pause for a few minutes. Somebody can please type in the chat box if you can see my screen as well as. All right, yes. So you can see as well as hear me. So what are we going to cover? We are going to cover what are the various biochemical reactions, what are the prerequisites, what are the how do you inoculate them, how do you read them, what are the principles, and what do you actually need to understand when you are asked, because biochemicals are usually asked as a viva question in your practicals, and they are essentially explain your 3 years work that you've done in your laboratory as to how closely you've done. So just don't listen to the lecture. Go back to your departments, try to make or see as many media as you can, and try to inoculate them with your own hands. Try to read them, their positive, their negatives, so that you understand the basic fundamentals. So what are the prerequisites for any biochemical reaction? First is a quality assured media, which has been checked with a positive and a negative control, and an uninoculated tube has also been inoculated as a control, so that's to show that the media itself is not contaminated. Then, of course, pure culture of bacteria, which means that just give me a second. Yeah. Pure culture of bacteria, which means there are single well separated colonies from primary diagnostic plate. If not possible, then subculture on a non selective media. And if you find that the culture is too contaminated, there are too closer colonies, and you will not be able to take a fresh colony, then you subculture in a broth or an agar slope, or you subculture on a non selective media if it is completely mixture, so as to separate the colonies. If it is otherwise, if you're doing it, it's always better to subculture in a broth, just like you do it for antibiotic sensitivity, or on an agar slope, and do it after 4 hours, or making a fresh suspension in the broth. Now, for which we make a fresh suspension, for which we inoculate, we'll be discussing along with the actual biochemicals that we do. Now, always remember, any of the biochemicals which are made, it is not advisable to autoclave them at 120 degrees, because autoclaving them is going to break down. For example, disaccharides are going to break down to monosaccharides and so on and so forth. So it's always better either to sterilize them by filtration or use a lower degree of heat for a shorter duration of time. Now, why do we pick up a colony from a non-selective media? This is a very common question which is asked in the exam, and the reason is very simple, that all the characters are expressed best when you culture a colony on a non-selective media. Only an isolated colony, you should know which controls are there. Very often, when you say that I'm going to put up the controls along with the biochemicals, your exam center will tell you We've put up the controls. You just tell us which positive would you like to put up for citrate, which negative would you like to put up for urease, so on and so forth. But an uninoculated medium, just like we discussed in my previous session on quality control of media, we should look for contamination of media, working of indicator, and whether our incubatory conditions are working well or not. Now, you will realize there are multiple indicators which are used when we talk about media. There is Andrade's indicator, which has a range of pH 5 to 12. You look at it, it's one of the broadest in pH range that it has. And it turns from colorless to a bright pink color as the pH reduces. Now, in bromocresol purple, it's a very small range, 5.2 to 6.8, from deep purple to yellow. We are talking about uninoculated to acidic. Phenol red, 6.8 to 8.4, reddish orange to yellow. Bromothymol blue, 6 to 7.6, which is green to yellow. And rhesus urine, very rarely used, pink to colorless. Again, a very broad range. So, what do we keep in mind here is most of the sugar media, you will have Andre's indicator, and the reason for that is to compensate for the wide range of acidity and that you're going to see. Now, how do we classify these biochemical reactions? We often classify them based on either just look at what an organism needs to grow and multiply. It needs a carbohydrate source, it needs a protein source, it needs and certain minerals, and to utilize everything, it has certain enzymes. So, most of our biochemical reactions for identification and confirmation are based on these. Either we check for carbohydrate metabolism, or we'll check for protein, or we check for these enzymatic activities. And then there are a few which have combined one or two, like a triple sugar ion test is going to combine H2S detection along with the metabolism of three types of carbohydrates. Then let's take them one by one. First test is the test which is used to distinguish between aerobic and anaerobic breakdown of a carbohydrate. Then there are tests to show the range of carbohydrates and related compounds that can be attacked. And then there are tests for specific breakdown product. If it breaks down glucose, what does it produce? Tests to show ability to utilize a particular substrate. If I remove everything else and give the bacteria only one particular compound, which bacteria can actually utilize only that? So, what do we say? We say there is an anaerobic media and then there is an aerobic breakdown. What is the difference between the two? Can you type the why when we say aerobic and when we say anaerobic? What is the primary difference? Just type the chat box. Did I get any answer in the chat box or not? No answers. Everybody seems to be sleeping. What do we understand by aerobic breakdown and anaerobic breakdown? The presence of oxygen and the absence of oxygen. Yes. Will the end products differ? Think about that. So, in what happens is that when you talk of oxidative, the molecules will undergo oxidation, sorry, in the presence of oxygen and with a lot of enzymes. In fermentative, sugar molecules can undergo change to produce acid and alcohol. And this can happen in the absence of oxygen. And we all know how alcohol is produced in the world. That is by fermentation of carbohydrates, which is a bacterial breakdown of these carbohydrates, which gives rise to beer. So, the initial compound here in oxidative may vary, but fermentative will definitely require a sugar. The end product will also be different. In the presence of oxygen, the end products are usually carbon dioxide and water, whereas in the fermentative pathway, they are usually lactic acid and ethanol. The enzymes are oxidative, and whereas here there are multiple enzymes lactic acid dehydrogenase, acetaldehyde, and ethanol dehydrogenase. So, now where have we applied this in our biochemical reactions. Somebody, let's have this. Why in where which test biochemical test utilizes oxidative and fermentative OF media? Great. So what happens is that we have an oxidative fermentation test, which is Hugh-Levson's test, which determines this ability. Now, what does it do? It distinguishes the gram-negative or sometimes even gram-positive aerobic to facultative anaerobic bacteria. So, what ferments? Enterobacteriaceae, Aeromonas, Vibrio, Haemophilus, Helicobacter. And what oxidatively breaks down? Acinato, Pseudomonas, Stenotrophomonas. We also call them as nil fermenters because they do not ferment, otherwise do not. Then even in staph, from to differentiate it from micrococci. Then sometimes the media is semi-solid and it gives an extra information in the form of motility and gas productions. But what does this media contain? For gram-negative bacilli, the media contains peptone, salt, phosphate buffer, an indicator which is bromothymol blue 1%, and a sugar, either 1% glucose, lactose, sucrose, or maltose. Usually, glucose is preferred for a simple reason that glucose is broken down by all the organisms, irrespective of whether they are lactose fermenters or non lactose fermenters or whatever they are. Now, there is a staph OF media which is used to differentiate from micrococci. Traditionally, it is made with bromocresol purple as an indicator. You cannot use the same media for same indicator and the same media as you use it for gram negative organisms. How do you do it? Now, you take duplicate tubes, separate them, duplicate, and to one tube, layer it up. In one tube, layer it up with paraffin oil. ये बनाना बोलते हैं पका के मतलब आटा मार के और तीन रोटी. So you inoculate it in duplicate and you stab the media 3 to 4 times halfway to the bottom of the tube. And to one you add sterile paraffin to create anaerobic conditions, something like this. And of course, what is the QC? The QC will be E. coli, which is a fermenter, and a negative QC could be Pseudomonas, which is an oxidizer. So the Pseudomonas aeruginosa will only produce yellow color in an open tube and not at all in a tube which is laid with paraffin because there is no oxygen available. Whereas E. coli, it just loves to eat its glucose, so it will eat it either way, whether it is oxygen is available or it is not available. So even in the paraffin covered tube, it will find that it has not grown. And you see this flaring of the media because it's E. coli. It has tried to move out from this, and that is why you see this. Here you barely see the stab because there is hardly any growth. Alkalogens faecalis is non-saccharolytic and there is no color change in both the open tube. So you will find that whenever the glucose is metabolized, the color is going to change to yellow. And this is how you will interpret it. That what you will write in your exam sheet is more important. Right now, so you'll identify the organism and then you'll say. But how do you interpret and write it? Remember to write it like this. Remember to write it that it is with the paraffin oil, what did you see? Without the paraffin oil, what did you see? And what it indicates. So interpretation is either oxidative, fermentative, or non-saccharolytic when nothing is utilized anywhere. Now, the coming on to the range of carbohydrates and related compounds that can be attacked, the very famous carbohydrate fermentation test. Now, this determines that the ability of an organism to ferment a carbohydrate which is in the basal media and produce acid with or without gas. Now, how does it do? We know this that all Enterobacteriaceae are glucose fermenters. Some Salmonella enterica species have got some sucrose, some ferment, some other sugars, some don't. And there is an older classification you will find, which is division is on the basis of various raffinose, mannose, to differentiate between subspecies of Salmonella enterica. Then Shigella group A are mannitol fermenters, B and C, D are not. Always remember, this is very simple way to find out in your exam. If Shigella comes in the exam, your biochemicals are gone. You have to know your biochemicals very, very well. Then Haemophilus ducreyi and Haemophilus species, Proteus vulgaris from Proteus mirabilis. So, how does it work? How does it work as a carbohydrate metabolism? Now, we all know that there are a few things which work for carbohydrate metabolism. Let's try and understand it with the help of a lac operon. So, what happens is that one thing is that I'm really sorry, I'm having a bad cold. You have a repressor gene, and then you have a beta-galactosidase genes, and you have a permease gene. The function of the permease gene is to make the lactose enter. We're taking this with the example of a lactose and not glucose, because glucose anyway enters every living cell and is metabolized. Let's understand this with the help of a lactose. So it has a permease enzyme by which the enters, and then lactose is broken down by beta galactosidase depending on the switching on and off of repressor to convert it to glucose. Once it becomes glucose, then it is metabolized by any organisms. So, what is the key indicator here is one is this permease and the other is this, which is going to convert it to either glucose or the one which is going to make the sugar enter. So there are various kinds of sugars. You will find that there is polysaccharide, disaccharide, monosaccharides. Everything ultimately is going to break down. So that is why we say while preparing a carbohydrate media, never heat it, because what will happen is lactose will break down to glucose plus galactose. So a disaccharide will actually break down to monosaccharide, and glucose is fermented by all. So you'll find that there is a faint pink reaction because the organism is able to metabolize glucose. So always remember that is the reason why we don't do it. What do we use for quality control? We use E. coli, Pseudomonas, and uninoculated tubes. The indicator, what do we use as an indicator? Most commonly, Andread's indicator is used, which turns reddish pink at about 5.5. We've seen its range 5.5 to 12. Now, how do you prepare it? You add NaOH 1 moles to a liter to a 0.5% solution of acid fustian unless the color becomes yellow. It's one of my favorite questions I asked in the exam. What actually is Andread's indicator? And the answer is so simple. It's acid fustian in sodium hydroxide. So it is used as a concentration, a very low concentration of 0.005% in the medium, and it fades very rapidly when it is stored. That's the problem of Andread's indicator. So if you have a sugar media which is lying in your fridge for 7 days or 10 days or for 20 days, the Andread's indicator loses its work. It may not function as good as it was in the fresh media. The other indicator which I used are bromocresol purple, 0.2%, where bromocresol purple dye is used to give a final concentration of again 0.005%. This doesn't fade, though it can be used. It is yellow in the acidic pH and violet purple at 6.8. Remember this. It is the reverse of Andread's indicator. Then we have phenol red, bromothymol blue. Phenol red, you all know, is used in so many media. It's actually phenol sulfathin, which is again used in a sodium hydroxide base, and it is dissolved by gentle heating, and then acid is added to it to make it neutral. It does not phase on storage, but it's very good for serum media because it takes very small changes in pH. It is yellow at 6.8 and is again purple pink at 8.4, just like bromocresol purple. So, what is it does is that it is very good if you're looking for fermentation by nutritionally exacting bacteria or in a serum containing media, it works very well. Then you have bromothymol blue, which is again made in sodium hydroxide and distilled water. It can be used as an alternate to phenol red in serum sugars when the pH changes are very small, and is also used in broth based media or in organic acid fermentation media. So it is yellow at 6 and is blue, as the name says, bromothymol blue at 7.6. And what is the medium? The medium is very simple. Either it is a peptone water or peptone broth, and a sugar of concentration of 0.5% is added to it. Android's indicator is the most commonly used, so I've written only Android, and then a reverse Durham's tube is added. Now, if you want to prepare serum sugars, one of the older lab technicians in media room will tell you, to this medium add a drop of serum or add a 1 ml of serum to each tube. That is the shortcut method, but it's not the actual way. Actual way is to have a peptone serum water which actually has 25% serum in it, and add a drop of sterile serum to peptone water. And then sugar is also double. Instead of 0.5, it is 1%. And the indicator here is phenol red or bromothymol blue, as I have explained earlier. How do you inoculate? You inoculate using a small pasture pipette or a small dropper from a liquid broth which has been incubated for 4 to 6 hours. Why do we incubate the broth and don't use colony directly into carbohydrate fermentation media? Imagine yourself coming home. The moment you enter and your mother says, "See here, have your food." What is your reaction? You'll say, "Oh, let me change, let me relax, switch on the fan, let me cool down. Only then will I be in a position to eat something." So the same thing happens to the bacteria. You pick it up from the colony and put it inside any biochemical media. It takes time to adjust, and it's that time actually is your time. That is the incubation time that is getting wasted. So you have to get your bacteria in some form of a log phase, as someone has actually pointed out. Thank you, Dr. Darshan, before putting it into a biochemical media. So the end product will depend on what your organism is, what is the nature of substrates, and what incubation you used. 48 hours, 24 hours. As you will we'll see later, in some media you will require 48 hours. So when you test for specific breakdown products, what are we concerned about? Now there are certain organisms which most of them break down to lactic acid and water, which further breaks down to carbon dioxide and water. Certain bacteria, in addition to doing that, what do? They form stable acid end product. And to overcome that, they produce so much of acid that the buffering system is not able to maintain that. So these acid end products are seen in all organisms at 18 to 24 hours. But what happens after 24 hours is the organisms start dying in that acid. They are not able to tolerate that much acid. But there are a few organisms which are your MR positive organisms which continue to produce these acids even after 48 hours and remain alive in that system. These organisms overcome the phosphate buffer system which is used in the media. You remember sugar media also we used a buffer K2H. Wherever you see K2H2PO4, it is a phosphate buffer system. So any of the organisms which can use an alternate product and produce neutral end products are MR negative, and those which continue to produce acid at 48 hours are actually MR positive. So what we use this to differentiate between these. So what is the media used? The media uses Clark-Lewis medium, or we call as the MRVP broth, or the glucose phosphate peptone water, which contains peptone, glucose, phosphate, your buffer system, and a water maintained at pH of 7.6. You see, the high amount of glucose is there because we are going beyond 48 hours. So we require the organism to be given that much of this thing. मम्मी कहती है ना एग्जाम से पहले अच्छे से have 2 parathas before you go so that you don't feel hungry. So it's the same situation. Or the indicator here is methyl red, which is used, which works from a range of 6 to 4.4, and you incubate this. Now at 48 hours, you add drops of this methyl red reagent. It's actually nothing but a methyl red dye, which is put in 95% ethanol. Remember, this works only when it is made in ethanol. So it is added directly to the broth, but you declare it negative only after 5 days. So it is not possible for you to do it in the exam, because nowadays the exams are only for 2 days. But earlier, when the exam was for 3 days, we were required to show this result. So when you add this and it still remains red, then is your MR positive. But if it becomes yellow, then it is MR negative. Sometimes there can be a delayed reaction, which is to be taken as negative. That after some time, this will become slightly orange pink, but it should not be counted. There is a micro technique also which detects MR positive organism in 18 hours, and a very small amount of glucose phosphate broth is taken here and a drop of MR reagent is added to it. Then what is a VP test or a Voges-Proskauer test? Here, the neutral end products are formed. Glucose, instead of going on for forming lactic acid, it forms acetone, and this acetone remains in this. Now, if you add something which is acidic, too much alkaline in nature, it combines with alpha naphthol, which acts as a color intensifier in this 40% KOH and forms a compound which is a diacetyl, which gives a pinkish red color. So, three things are happening. From the what are the reagent you're adding? You have the same glucose phosphate broth, same way of inoculation. After 48 hours, what do? If your MR has come negative, now either of the reagent you add alpha 5% alpha naphthol in absolute alcohol, or you add it in 95% ethanol. That's how we make it. And then there is O'Meara's reagent, which also contains creatine. So what it does here is alpha naphthol and acetone combine to form a diacetyl, and it picks up a guanidine nucleus which is present in the peptone or from the amino acids which are there in the peptone to give you a very large pinkish red color. The sorry, the picture is not there. Yes, the picture is there. It's not like this. It's slightly different color. It's a very darkish red color which spreads soon to the complete medium. So they should be done in conjunction. Very often you will find that there are a few exceptions. Otherwise, if it is MR positive, it is VP negative. If it is VP negative, it is MR positive. Or if it is VP positive, it is MR negative. So the precaution which has to be taken here is that you have to shake it gently and allow it to stand. Usually, students get worried, they don't do this step, and then they get it a negative color. Once you shake gently, is that the little amount of heat that produces accelerates that reaction, and you allow it to stand, then it combines with the peptone and it gives rise to just then VP positive is pinkish red in color, VP negative is yellow. And there is also a rapid VP test, just like we had a rapid MR, where you inoculate tiny amount of broth, 0.2 ml, and these creatine. Wherever you are seeing these micro elements, that's what has given rise to all the automated systems. That these tests are done in the automated systems in a miniature manner. So there are very few organisms who are both MR and VP positive, and all PGs should have a list of that. Vibrio cholerae, Eltor, Aeromonas, all Listeria, Vibrio meshcomi. Pseudomonas uses the enterodox pathway to form pyruvate as an end product from glucose. So there is no glycolysis. So it is both MR and VP negative. Please remember this. Very often you people get confused and you say that Pseudomonas is a nil fermenter. It's not because it is nil fermenter. It is because its end products are very different. And those end products cannot fit in your reaction, which you are doing here. Now to show the ability to utilize a particular substrate. Citrate utilization test. Citrate, where does citrate comes from? Citrate may be utilized by bacteria via a TCA cycle or a citrate fermentation pathway. Now this citrate fermentation pathway breaks down to oxaloacetate, which is to pyruvate, and then to acetate, formate, lactate, acetate, acetone, and carbon dioxide. Now, what does your knowledge lie? You have to understand that there are Siemens citrate media, there is Courser's media, and there is a Christiansen citrate sulfide media. What are the difference between the two? Siemens and Courser are essentially similar, only that Courser has no agar and there is no indicator. So it's a liquid media, and you see its positive positivity by turbidity. Cement citrate contains sodium citrate, which is an ammonium dihydrogen phosphate, which is again a phosphate buffer. Sodium citrate is 0.2%. There is salt, agar, and the indicator is bromothymol blue. In contrast, Christensen agar contains 0.3%, almost similar, but additionally it contains glucose, which is not there here, and the indicator is phenol red. So it tests for citrate in the presence of organic nitrogen. There is a H2S detection system also. So Christensen citrate is actually sulfide medium is actually doing three things. It's detecting H2S, it's checking citrate in presence of glucose, which so in cement citrate, glucose is not there. So you check for actual. That is why cement citrate is used more common than Christensen citrate. What is the procedure? You all know citrate. It's blue in color. You do a light inoculum from a straight wire in a fishtail manner or a slant. Positive is Klebsiella, Enterobacter. Very common. Negative is E. coli, Shigella. And now metabolism of protein and amino acids. Metabolism is, we'll check that it is, sorry. So one of them is indole test. What does it check? It checks that how tryptophan has been split by the organisms. And this is a tryptophan or a peptone broth is taken in which tryptophan should be there, or you can take casein medium, which anyway. Remember last time I told you in my class why do we prefer what is casein and how does it help? Because it contains tryptophan. So if you add your indole reagent to it, which is nothing but alcohol and a compound which is known as para-dimethylamino-benzaldehyde, or an Ehrlich's reagent which is ethyl alcohol and contains the same compound, what it does it combines with that the reaction which you see here, it combines with the indole pyruvic acid and gives you a bright red ring. Sometimes some reagents you need to add ether so that the ring comes up. In some you don't want. Then there is a spot test which you can do. Filter paper strip, which is with Covax reagent, which converts brown to red. You can also do it for anaerobic bacteria in a similar manner, but use some acid. Then you also have a micro technique. As I said, for all biochemicals, we have a micro technique. Then there are multiple combined tests. You have an indole nitrate, motility indole, you have motility indole ornithine medium, sulfide indole motility medium. Now coming on to an interesting test, which is amino acid decarboxylase and arginine dihydrolase. Now, how does it work? There are two enzymes which are there with bacteria, decarboxylase and dihydrolase. Decarboxylase is decarboxylate lysine and ornithine to cadaverine and putrescine respectively, and arginine is dehydrolyzed to citrulline and ultimately to putrescine. And this is very useful in Vibrio aeromonas, differentiating them, or for certain Shigella, certain Salmonella, Klebsiella. So all these it helps. What does it have? It has a molars decarboxylase broth, which contains peptone, beef extracts, pyridoxal, glucose, water, and amino acid, whatever amino acid you have. The indicators are two here. Bromocresol purple, which works from 5.2 to 6.8, and cresol red, which works from 7.2 to 8.3. Now you want to have three distinct kind of colorations. Amino acid will metabolize, giving rise to ammonia, which is going to increase the pH. But there is glucose in the medium. So once the bacteria utilizes glucose, the pH is going to come down. So to detect all these minor changes, you have these two indicators. So once you've inoculated, always remember that decarboxylases are inactivated in low pH. So if that is the reason why you have a glucose here. So the organism starts multiplying glucose, forms lactic acid, which lowers pH, and then only decarboxylase will get activated. So that is the reason why you have these two indicators. So in 18 hours, you will find, 18 hours, if you check, you will find that the color has changed from light purple to yellow because the glucose has been metabolized and the pH is somewhere borderline. At 24 hours, the color will become purple again. Why? Because the pH has started now to increase beyond neutral and will become violetish or cresol red, purplish by becoming more alkaline. So you have these four tubes you inoculate. That is, one contains your L, the other contains A, and the other third contains O, and you have an uninoculated tube, which we call as the control tube. And all these tubes are layered up with paraffin so that you will find that they are all working under anaerobic conditions. These are the control organisms that I used. The alternatives that there are multiple alternatives. You can have a lysine ion agar. You can use Nestler's reagent, which detects ammonia production by arginine breakdown. You can have a ninhydrin broth. Now coming on to tests which are based on enzymatic activity. For example, urease. Very simple. The urea is broken down to uh, ammonia and carbon dioxide, and by the presence of urease enzyme. There can be Christensen's urea agar and the Stewart urea broth. Both use phenol red. Base should never be heated because the heat will decompose the medium. A long slant or a short butt can be prepared. Even liquid media can be prepared. And there should be a heavy inoculum. Do not stab the butt. You just fishtail inoculate something like this. So Christensen's agar, there are rapid just, and this is what you will see. You can also use urea strip test, urea disc test, and urea tap. Now, what are phenylalanine deaminase malonate? As I said, there are certain miscellaneous tests which are combined tests. Now, this determines two things. One, to deaminate phenylalanine, done by Proteus, and also to check malonate utilization. So, you prepare a broth medium which contains bromothymol blue as an indicator. There's a heavy inoculum. Malonate converts dark blue to light green, and phenylalanine, which you add as a ferric chloride solution, which combines with this, giving you an intense green color of phenyl pyruvic acid with ammonia. This is how it looks like. The malonate will look blue, whereas phenylalanine. So you can do this as a combined test. Coming on to a few reactions like the Catalase peroxidase test, everyday bench test which you do detects the ability of the organism to break down H2O2 to water and oxygen. There is a slide method. Now, with an inoculating needle or an applicator stick, put some colony on the slide and add a drop of H2O2. Usually, you do it the reverse, but this is how technically it should be done. In a tube method, you add it directly. You can also pour it directly onto the media and you will see bubbles there. But the thing is, don't do it with blood agar. And remember to subculture it first because it's going to kill the organism. There is a superoxole 30% test which is used, and then there is a mycobacteria differentiation test which is used along with tween 80. Mycobacteria will require tween along with 30% peroxidase solution because if it's cell wall which has mycolic acid and will not allow the enzyme to work. Then there is a whole cell peroxidase catalase test. There is a rapid semi-quantitative test for Enterobacteriaceae which can be used for immediate response. You know, catalase positive organisms, this is the list. All these lists should be there with you that, strep, entero, these are all negative. Oxidase is again cytochrome oxidase, which activates the oxidation of cytochrome. Molecular oxygen, which acts as an electron acceptor in the terminal stages. This helps to identify all the oxidase positive organisms like Pseudomonas neisseria from the Enterobacteriaceae. This is the list of oxidase positive organisms which is there, and it helps you to identify these right at the bench. The reagent is a multiple reagent. Most commonly used reagent is the Kovacs reagent, 1% tetramethylene paraphenylene diamine dihydrochloride. It's tongue twister, but you have to remember it. Solution is colorless, test toxic, more acidic. And then there is a modified oxidase test which contains DMSO. And this is often asked in the exam what is modified oxidase and where it should be used. What is the procedure? Fresh solution can be poured on the medium. and the colonies will turn purple in about 10 to 15 seconds. But subculture should be done fast as it can kill, because it's after all a reagent which can kill the bacteria. There is a dry filter paper method and a wet filter paper method, something which you are very familiar with. You do it every day in and day out. And then there's also an oxidase discs which can be used. You can do swab test on this, and there is a modified test which is used for differentiating between micrococcus and staphylococcus. This is how it looks like. Now, nitrate reduction test. One of the definitions of enterobacteriaceae is nitrate reduction to determine the ability of an organism to reduce nitrates to nitrites, and it further can become into and finish off in the as a gas. So remember, that can also happen. So what are the reagents? Now don't confuse these with VP reagents, please. That is alpha nephthol. This is alpha nephtholamine. It's a carcinogenic reagent, so please handle it very carefully. And nowadays we use an N-dimethyl alpha naphthalamine, because alpha naphthalamine is not very safe. Second reagent is 0.8% sulfonilic acid, and 1 ml of each of these to the test medium in 1 and B order. And you'll find that if there is a red color within 1 to 2 minutes, it's positive. If there is no red color, then it's considered negative. Then you add zinc dust, and if you still see no color, then it is just, but means that it has further reduced nitrates to nitrogen gas. Nitrate reduction test is also done for mycobacterium, and a commercial nitrate strip test may be used. This is how nitrate reduction test looks like. Now, detecting H2S production. H2S production, how can you detect? H2S production can be detected by viewing a visible black reaction. So you can use what is most commonly used is a triple sugar ion test. There are further many tests which I use: Kigler ion, sulfide indole, peptone ion, and so on and so forth. Lead acetate paper is also used at some of the places. So what does triple sugar ion do? Triple sugar ion contains lactose, which is 1%, glucose 1.1%, and whereas triple sugar ion contains lactose, sucrose, and glucose 1, 1, and zero point 0.1. Remember, glucose is less, because if you continue giving glucose to the bacteria, the sucrose and lactose will never be utilized. Indicator here is phenol red, and H2S indicators are ferric ammonium citrate and sodium thiosulfate. So you are all familiar with this. If you see a K by A, means that the butt slope is alkaline, but the butt is acidic, red by yellow, or it is yellow by yellow. Glucose is fermented, and lactose and sucrose is also fermented. Whereas if you see both red or alkaline, that means nothing has been fermented. And if you see an H2S streak, then it will appear as a black streak along this. Now, this splitting of the media, bubbles in the media, indicates gas production. Yellow button slant indicates that the organism produces huge amount of H2S. Now, beta-galactosidase test, which is to demonstrate the absence of beta-galactosidase. Now, remember, there was a permease enzyme and a ONPG, the beta-galactosidase, which was breaking down. So, the lactose fermenting bacteria need to have a permease for bacteria to enter. Otherwise, they will be late lactose fermenters, because their permease is not working very well. So only when the bacteria gets concentrated by a huge amount of lactose is it able to enter. ONPG does not require permease to enter the cell, so it identifies the lactose fermentation. You can use tubes, you can use one slide has gotten missed. So yeah, here it is. So what you do is you inoculate the peptone very, very heavily and incubate it for 18 to 24 hours. You centrifuge and discard the supernatant and add a drop of toluene, and then keep it in a water bath and add this solution. And then you re-incubate for a few minutes. A positive is indicated by yellow color, and negative is indicated colorless even after 24 hours. So remember, these are the two ways of doing it. Nowadays, you get ONPG discs, which I have it, I think, in my slide of photo. No, yes, you have test tablets. So within 6 hours, they give you strongly positive. They are filter paper impregnated discs. So similarly, you have a PNPG test, which is used for mycobacterium also. So I think with that, I will put an end to this year's class. Now the next class will be on 6th with microbial genetics from Dr. Priyanka Narayan will be taking it from Patna, and the next one would be on 27th where newer advances in sterilization disinfection. We'll let you know and circulate whenever we have this. So thank you so much. If there are any questions, please let me know. Thank you for joining, all of you. Hope you had a good learning experience. My apologies, I'm not well today, but I had to take the class, so I've tried my best.